Good afternoon, everyone. So it is my pleasure to introduce our distinguished IAS lecture speaker, Professor John Hardy. So we would like to thank our friends, John, Henrik, Amanda, and Marion, to come to Hong Kong again after COVID. So John is a well-renowned neuroscientist and is known as the father of genetic study in Alzheimer's disease. So he started his work on this neurodegenerative disease in the 1980s. Indeed, at that time, when the academic world had little knowledge of this disease. So in his early career, he and his team discovered the known cause of Alzheimer's disease, a genetic mutation of the amyloid precursor protein. Based on this exciting finding, he and his colleagues devised the amyloid cascade hypothesis. So this significant finding indeed is formed the basis for the nearly all basic and also clinical research in the disease, including the recent approval of the A-beta antibody for the treatment. John has also conducted groundbreaking genetic research on other neurodegenerative diseases. And his outstanding research has resulted in over 1,100 scientific papers with more than 100,000 SCI citations. So no shoe. He is the most cited Alzheimer's researcher in the UK and is a fellow of the Royal Society and member of the Academy of Medical Sciences and the French Academy of Neurology. He has received numerous prestigious international awards, such as the Breakthrough Prize in Life Science in 2016 and the Brain Prize in 2018. He is also awarded an honorary doctorate by HKUST in 2020 for his distinguished research achievement. John is currently the Professor of Neuroscience at University College London and the Senior Investigator at the UK Dementia Research Institute. He is the IAS Senior Visiting Fellow and has a long-standing relationship with HKUST. He is a team member of the Hong Kong Center for Neurodegenerative Diseases and Inno Hong Kong Center at HKUST. Together with the visitor here, so we are conducting innovative and pioneer research to uncover the causes of Alzheimer's disease in our center. So we are honored to have Professor Hardy with us today to present his talk entitled "The Genetics of Major Neurodegenerative Diseases, from Mechanisms to Treatments." Please join in welcoming Professor Hardy.
And I mentioned that when we do a GWAS, we have to do a bumper-only correction because we're testing the whole genome. What about if we reduce our bumper-only correction just by looking at those genes which respond to amyloid? And so that's what we did, and this work was led by Dervis here. And you're looking at this response network again. Here's TREM2 again. This is the same network, it's just in more detail. Here's TREM2 at the middle. These are the genes we've already been discussing. And these are the genes which were independently associated with Alzheimer's disease. I'm not going to have time to talk about these genes individually. I'll just mention a couple because they're of interest and because they relate to conversations we've had this morning with the group here. This gene SPI1, this is a microglial master regulator gene. It's a gene involved in coordinating microglial response. This gene here, CXCL10, is an amyloid response excitatory cytokine. It's also one that's shown up in networks that were shown to me this morning here at HKUST. This gene here is one we are continuing to work on, OAS1. Dervis is working on this gene. this is part of the interferon response. And it's of interest because the same gene, in fact, the same haplotype, is involved in the COVID response. So it's not involved in determining whether you get COVID, it's involved in determining how badly you get it. Once you've been infected, are you going to have a bad response or a good response? And it's the same gene, OAS1, is involved in bad COVID. It's a part of the interferon. It's part of the interferon response. So what it responds to, by the way, is at least one of the things it responds to is free DNA. It responds to free DNA, so it's for example, therefore part of a viral response. So if you have if a microglia comes across DNA fragments, this is part of the response to get rid of those fragments. So I'm not going to talk about these more than that, just to say that much. We've just completed what I think is a really nice set of experiments looking at brain slices in the mice to see when the genes change in their expression levels. And what we see is that the TREM2 response is only when the microglia touch the plaque. In other words, if you've got a mouse depositing amyloid, the only microglia which upregulate TREM2 are the ones which physically touch the plaques. The microglia which are away from the plaques don't upregulate. And there's a whole series of genes. This is all published, so you can look at it online. A whole series of genes which upregulate on contact. There's a fewer genes which upregulate more distant from the plaques. Presumably, for example, cytokines are responsible for this distant upregulation. So this gives you more microscopic detail of when the upregulation occurs. And we think we know why this is, and that is because, in fact, TREM2 is a phospholipid receptor. So in other words, TREM2 upregulates when it binds phospholipids. That's when it upregulates. This very nice paper from Marco Colonno makes this clear. And a gene I haven't really mentioned, but it's also an Alzheimer's risk gene, ABCA7, is a phospholipid uptake site. So what these two papers together started me to think about what could be the mechanism, what could be the relationship between the autosomal dominant Alzheimer's disease, which were all involved in APP processing and amyloid production, and these genes, which are all phospholipids and microglia. And this is the idea that I came up with. And this is how I drew amyloid metabolism before in that slide much earlier on. But this is what I actually think happens. I think amyloid deposition begins in the membranes. I said how difficult I thought it would be to get amyloid outside the membranes. I think the deposition begins in the membranes and starts to disrupt the membranes, and that what happens at the beginning of Alzheimer's pathogenesis, what happens at the beginning is that ApoE and microglia deal effectively with that damage. And it's only when that damage overwhelms these protective mechanisms that the pathological cascade continues. In other words, in the first stage, you get amyloid deposition occurring within the membrane, but that's okay at first because the microglia and ApoE carrying the lipids and the amyloid together deal with it without it causing further damage. So I think this is what happens. And consistent with that is this genetic analysis that we have done in collaboration with Valentina Escott-Price. And what we've done here is compare the GWAS for Alzheimer's disease, which I've shown you, with the GWAS just for amyloid deposition. So we've now got many samples, for example, in ADNI, where we have got the PET scan results. We know there's amyloid deposition. So what is the risk of that amyloid deposition? And the risk of that amyloid deposition, is almost entirely ApoE. But the risk for dementia is contingent, of course, first on the amyloid deposition, but then on the rest of the GWAS, which is microglial. So it too is dividing the pathogenesis. Amyloid deposition, nearly all ApoE. Dementia in the context of amyloid deposition, ApoE therefore, plus the microglial response, a two-stage process. And this is a beautiful paper, I think, from David Hansen's group. And what he has done in this paper is he has repeated the experiment that we had done, where we crossed the amyloid mice with the tau mice. But now he's done that experiment when he's paralyzed the microglia by knocking out the TREM2 gene. So he's done the same experiment, amyloid tau, but in the context of disabled microglia. And the tangle pathology is much worse when you disable the microglia. So what this paper is saying is microglia, the microglial reaction is protecting you from the downstream effects of amyloid deposition. Of course, that protection wears off, but that's how it starts. I should say, I had too much soy sauce with my my lunch, so I'm getting very thirsty. I would have had some beer, but I knew I was lecturing, so it's your fault. I had soy sauce and no beer. So this is my summary for Alzheimer's disease for now. Production is largely amyloid. Early onset disease is largely amyloid production. Some of the loci, I mentioned ADAM10, are involved in metabolism, but most are microglia, and the vast majority of the genes are amyloid responsive. And we now know that many of them are involved in amyloid clearance. And I have suggested, it's only a suggestion, that this relates to disrupted membranes. Now, I promise much more quickly, I'm going to go through Parkinson's disease. This is the pathology of Parkinson's disease. This is a Lewy body, electron micrograph of a Lewy body. I think, by the way, I think of a Lewy body as being a failed lysosome. That's my simple-minded view of it, as a failed lysosome. Here, anyway, you can see damaged mitochondria within the Lewy body and many, many, many membranes. Now, synuclein is an amphipathic protein. Amphipathic means there's helixes, one side of which are hydrophobic and the other side of which are hydrophilic. So an amphipathic helix lies along membranes. So what you can suppose is that these membranes all have synuclein, if you like, stuck to them, basically. So you can see then damaged mitochondria and lots of membranes. What happens when we look at the genetics of Parkinson's disease? Well, we see essentially two types of genes. Endosome lysosome genes and mitophagy genes. Mitophagy, I've just shown you damaged mitochondria, so that fits completely. Endosome, lysosome, I've just mentioned GBA. GBA is perhaps the most prevalent risk locus. Homozygous mutations in GBA give you the lysosome storage disease, Gaucher's disease, and heterozygous GBA mutations predispose you to Parkinson's disease. So mitophagy and the endosome lysosome system. So this really is consistent with the pathology. I showed you the Lewy body with damaged mitochondria, mitophagy, and lots of membranes, probably endosome lysosome membranes. So the pathology fits with this completely. When we do the GWAS, obviously I'm not going to go over this, but what we see in the GWAS is many, many lysosome genes and many mitochondrial and mitophagy genes. So the same pathways coming again. How do we fit this together? And I think this paper is the best clue for that, because what Dimitri Krank says is that synuclein is metabolized through the lysosome, and I showed you all those membranes inside the lysosome which will be coated with synuclein, and that if you overexpress synuclein, then you inhibit lysosome flux. So I'm going to make the same argument as I made for amyloid and microglia. I'm going to make you the same argument for synuclein and lysosomes. So, in other words, in Alzheimer's disease, I've said amyloid metabolized through the microglia, and in Parkinson's disease, synuclein metabolized largely through the lysosome. And then finally, thinking about the tangle diseases, what we find with the tangle diseases, the top hit I've mentioned already is tau itself, overproduction of tau. We don't understand all of these genes, but what we do see is the ubiquitin proteasome system as risk loci for tangle disease. And in fact, we now know that one of the major routes by which tau is metabolized is through the ubiquitin proteasome. So I'm making the same argument again. Tau overproduction, or if you can't metabolize it, and lack inhibition of clearance. So the same argument. So I've made the same argument, if you like, 3 times, only with different proteins having different degradative pathways, and that gives rise to those different pathologies. Now, one of the most puzzling things that we face over and over again is especially we geneticists and molecular biologists, what we do is we talk about these diseases as if they're pure diseases. But in fact, anybody, the epidemiologists and the neuropathologists, always tell us that you almost never get pure pathologies in these elderly individuals. Most elderly individuals who die, when you look at their brains, they've got multiple pathologies, not just plaques and tangles, but they've got plaques and tangles with Lewy bodies, with TDP-43 pathology, and so on. So you don't see these pathologies as pure pathologies. Why not? Well, I've suggested this: amyloid, microglia, tau, ubiquitin proteasome, synuclein, lysosome. But of course, that's a massive oversimplification. And we know that there's crosstalk between all of these clearance pathways. So there's crosstalk between these pathways. And so if one of these pathways starts to fail, then it's inevitably going to push the other pathways into failure as well. And so you don't have to postulate a direct interaction between amyloid and tau. What you do is you postulate that your clearance pathways are close to failing, and if one of them does fail, it causes the others to fail as well and lead to the other pathologies. So this is what I think of in terms of mixed pathologies, that you're seeing a sequential failure in the different major clearance mechanisms. Now I'm going to talk briefly about the recent, if you like, breakthroughs in terms of treatment. This is the first paper. This is the first paper looking at amyloid antibodies and clearance. And I just mention why this work was done. At the time, it seemed so curious that anybody would try amyloid antibodies or immunization. And in fact, the reason it was tried is a perverse reason. At this time, there was a series of papers from Pat McGeer in Vancouver and Joe Rogers, who was then in Arizona, pointing out that when you looked at plaques, you saw a lot of the complement cascade at the plaques. So showing, therefore, that the complement system was activated around plaques. And so Joe Rogers and Pat McGeer gave talks at Athena, now Elan, or then Elan, gave talks at Elan about this activation of the complement cascade. And at the time, we had a conundrum, and that is we've been able to make mice with plaques, but those mice never of themselves develop tangles. And so what the Elan group decided to do was to try and, if you like, bump up the immune system, so that if they could bump up the immune system, maybe the amyloid plaques would cause tangle formation. So in fact, they'd done these immunization experiments to potentiate tangle formation in mice which were already making plaques. That was why they did the experiment. But what the result they found was that the amyloid immunizations cleared the plaques. So it was a good experiment, but an accidental outcome of that experiment. Now, what they found, though, the experiments in mice, all of the early experiments in mice, were given in mice before amyloid buildup had started. And what the experiment showed is that amyloid immunization prevented amyloid buildup. These early experiments did not show amyloid clearance. What they showed is the prevention of buildup. And that really, all of us in the field really misinterpreted those results. What the experiment showed was they prevented buildup. The only time that you saw clearance in these early experiments was when you got blood vessel damage. And when you got blood vessel damage, very high concentrations of the antibody went into the brain, and where that happened, you got clearance. That's what these early experiments showed. And these early immunization experiments in humans were not successful. The immunization and the giving of antibodies were not successful, and they weren't successful because they did not remove amyloid from the brain. And that changed with these later experiments. First, well, we can talk about the first, but lecanemab, the first really obviously successful experiment. And here are the other ones. Aducanumab, which was arguably successful, and then donanemab as well from Lilly. I'm just going to show you these data. What you see in this data here, in this 18-month trial, is when you give lecanemab, by about 6 months, you've really reduced on amyloid PET. So you really see, if you like, that the antibody is sucking amyloid out of the brain. And consistent with that, you can see that it separates the clinical outcome is favorable. Two things. It's definitely favorable. There can be no, I mean, there is some argument in the literature. I think that argument is crazy. It's clearly successful, but it clearly does not stop the disease either. It slows it, but it does not stop the disease. And donanemab is essentially simple. Now, the reason this is, if you're going to read a paper about this, in my view, this is the paper to read from Eric Karen and Bart de Strooper. And the reason I like this paper is because it's written before these trials. So it predicts which trials are going to work. And a lot of people are clever after they've seen the results. Everybody's clever after they've seen the results. The reason I like this paper is because it was written before the results. And what they suggest is when you look at when you're looking at trials, this is what you should expect. If a trial is going to be successful, it has to go on long enough for amyloid to be reduced and then to continue for long enough for you to see a clinical effect. So the trial has you can think of it as two phases. The bit of the trial where you're sucking amyloid out and then running it long enough so you can see the separation between cases and controls. Now these are the real data from all of the trials, and now I've shown you that theoretical trial. This is the real data. Here you can see donanemab and lecanemab. These are two trials everybody, apart from a few crazy people, agree has worked. Now here's aducanumab. Aducanumab was really a trial where there was a lot of controversy. It was approved in the US, but in fact, a lot of people, actually including me, felt it should not be approved because there were two trials. This trial worked. This is the amyloid reduction, and this trial did not work. And so this trial also makes sense in terms of the predictions that Karen and de Strooper had made. And then this trial was announced late last year by Roche. And yeah, this trial failed. So everything fits with the de Strooper and Karen model being accurate. And that's important because it tells us that any future trial for anti-amyloid drugs in Alzheimer's disease, we know what we need to do. It's given us something to shoot at. We know what we need to do. Now, Now I'm going to talk about what I haven't talked about. I should have put a slide in about it, and I'll have to talk without a slide. There is a serious side effect that both lecanemab and donanemab have, and that is this ARIA, amyloid-related imaging abnormality. And what that's usually, what it is, listen to the name, amyloid-related imaging abnormality. For many people who are on the drug, if you look at an MRI, you see an inflammation. In most of those people have no clinical symptoms they're aware of. But if you look at their brain, you can see there's an inflammatory response. And in a few of them, or a percentage of them, they have headaches. So that's the majority. Very occasionally, you get those individuals develop hemorrhages. And if they're on anticoagulants especially, those hemorrhages can be fatal. But at each stage in that process, it's a few smaller and smaller percentage. What's causing that? Well, what's causing it is very simple. It's the antibody hitting the amyloid in the blood vessel. So the first thing that the antibody hits at very high concentration is the amyloid in the blood vessel. And it causes an inflammatory response as it removes that amyloid. So we call it a side effect, But actually, it's in a way, it's not a side effect. It's the antibody doing its job, but on the blood vessel amyloid. And that's the problem. Now, can we get better? Can we, I mean, these drugs are going to be expensive. They're going to be expensive. And if you have to keep doing safety monitoring by MRI, that effectively doubles the price because you have to pay for neuroradiographers and neurologists to both administer the drug and test for safety. Can we get better? Well, I think we can get better. And I think we can get better mainly with the current drugs by getting better at diagnosing early. If we diagnose earlier and accurately, then of course the people might have less amyloid in their blood vessels, so less amyloid for the antibody to hit. But of course we'll also hopefully be saving more cognition as well. So there's a big imperative to get better at early diagnosis. Can we do it? I think we can, and in fact that's one of the reasons that we're here with Nancy's group at the moment. Can we get better? Well, we can. We can do genetic analysis to do genetic prediction, and that genetic analysis is reasonably good. It has an area under the curve of about 0.8. It's not by no means perfect, but it helps. We can get better. And here, this is work, in fact, from Henrik here, about using blood-based biomarkers for Alzheimer's disease. So one can imagine a situation where we're doing both genetic and blood-based biomarkers to get better at diagnosing early. There's still some way to go, but we are, I think, on the way to getting better and earlier diagnosis. One of the things I should say is that this analysis is based on Europeans. It's not based on Asians. We've got further to go to get a better polygenic risk analysis in Asians. We've just started to do that here in Hong Kong. And, you know, we have to get better. We've started, and we've just also started by looking at African populations, but we've got further to go. How else can we get better? Well, of course, the other thing we can do is start targeting also other treatments. And this is a paper from earlier this year from Kath Mumry at our place, looking at tau antisense. So obviously, one can imagine doing anti-amyloid antibodies and maybe tau antisense as an additive therapy. So that's another way. By doing combination therapies, we might get better. So this is where we are up to. Looking to avoid ARIA, earlier diagnosis, we need to organize. I don't know how it would work here in Hong Kong. I don't know the healthcare system enough. But in the UK, we need to get better at organizing dementia services so that we can routinely pick up Alzheimer's disease. What happens in the UK is that somebody, let's say a man of 70, his wife, I'm 69, so next year I'll be choosing a man of 71. His wife will take him into the GP and say, I'm getting worried about my husband. Yeah, it's getting close. And the GP will say, well, let's have a look at you now, but come back in a year's time and let's see how you're doing, or let's come back in 6 months. That's going to be no good, you know, because a year's time might be too late for these treatments. So we have to get better at really making the diagnosis earlier. And, you know, I might be taken into the GP the second time, and then the GP will refer me to a neurologist, and my wife is a neurologist, by the way, and so I might skip the first step. But, you know, the whole process of diagnosis could take 2 or 3 years. That's no good. That's no good. We need to make the diagnoses early and accurately. There's a lot of reorganization of the healthcare systems are going to be required. So that I think is the big imperative. In the UK, we've just set up a dementia challenge grant, which Amanda and Henrik are a big part of, doing biomarker analyses to try and get earlier and accurate diagnosis using blood-based biomarkers. So that's something that really you need to be thinking about here in Hong Kong. So where are we up to? Well, this is where I think we're up to. You know, in 1903, the Wright brothers showed on a North Carolina beach that they could fly a heavier than air machine for a mile. And in 1903, who would have believed that just 15 years later that we would have commercial flights and flights over the Atlantic? In other words, we've just achieved the first treatments which work at all. I think from now on it will get better. We know what we need to do. We need to, if you like, suck amyloid out of the brain. And we know that if we do that, we have a very high likelihood of success. So we know what we need to do. That's bringing drug companies back into the area. So there's more investment in the area again. People have begun to give up on Alzheimer's research, thinking it was never going to be possible. This is showing that it is possible. So yes, these are small steps, but I think we are now on the way to mechanistic treatments in Alzheimer's disease. And I think thinking about the other diseases that I've mentioned, I think the fact that we now know that if we target the pathogenic mechanisms, and we understand the pathogenic mechanisms, there are ways, even for those other diseases where we're a bit further behind, that we can develop treatments for those as well. Thanks very much. Happy to take questions, by the way.
So, questions from the audience?
By the way, I'm away for over a week, so I'm here for over a week. So if anybody wants to see me and to have, I don't know, a tutorial or whatever, I'm going to be around HKUST for the next week and a bit. So don't be shy. I'm happy to talk to people in smaller groups.
So can I ask a question? Thank you very much for impressive talk. So I liked the idea, including the one you mentioned that amyloid beta is produced within the membrane. Probably this kind of idea is coming from TREM2, acting as a phospholipid receptor, and also ApoE4 may change the delivery. So two related questions. One is, did you see some kind of membrane composition change?
No, but we've never looked.
Ah, I see.
We've never looked. You've got someone here who can look, so, you know, maybe you can start thinking about that type of research question here at HKUST. No, we've never looked.
Okay. Related another question. If it is changed, if really intramembrane production of A-beta plays significant role, then why it's happening? So maybe TREM2, maybe receiving some lipid, phospholipid, perhaps membrane fluidity will be different, so that the fluidity affects the exporting the A-beta 42. Just I imagined. So do you have any idea how to link the phospholipid towards the outcome?
No, although in fact we've been doing some experiments a little bit back in London last year, looking at changes in lipid composition actually in the CSF by ApoE genotype. We've just started to do it. We're still doing the analysis. So we have started. There are some papers from the 1990s on membrane fluidity and Alzheimer's disease. One of the authors was called Mick Bramner, B-R-N-M-E-R, So there was about half a dozen papers in the 1990s about membrane fluidity and Alzheimer risk, but nothing in the last several years.
I see. All right. Thank you very much.
I could just make a comment in this context too, Henrik Zetterberg here. The paper that John mentioned from Lucia Guerres-Chava's work with Bartos Trooper also indicated that there is a temperature sensitivity in the gamma secretase processing of APP. So if you increase the temperature, that also increases the fluidity of the membrane.
Yeah.
And then the gamma secretase is a little bit destabilized and lets go of the substrate earlier, so longer forms are released or at least not cleaved further. So it seems like fluid membranes will be a little bit bad from an APP processing perspective, given longer A beta forms. So it's and there is a link also with ApoE and the cholesterol-rich lipid drafts. That perhaps you also would like to comment on this.
Yeah, I think lipid handling obviously is tremendously important. So lipid droplet, as you know, is found in aged human animals. And I think E4 is a large pathway outside of seeding amyloid that's related to lipid metabolism. But if I can ask a question, John. So you talk about genetics, and I think it's clear those risk genes are either in the amyloid pathway or microglia or response lipid. What about the protective gene or gene variant? So, as you know, ApoE2 is protective, and they're a protective variant. APP has a protective variant.
Yeah.
But are there other genes or variant? Because I think maybe they can also give some clue about what pathways are important or giving a resiliency.
My favorite, in a way, my favorite target is PLCG2, where there is a protective variant which has a population in Europeans of about 3% from memory, and that's protective, and that seems to be in the TREM2 pathway, but downstream of TREM2. So but I agree with you. I think it would be great to find more of those protective variants. Incidentally, just Henna Holstiger in Amsterdam, I don't know if you know her, she has collected centenarians in Amsterdam who are cognitively normal. So she collected people over 100 who are still living at home and looking after themselves. And amazingly, the eldest woman in her collection is an ApoE4 homozygote, amazingly. But that woman has the PLCG2 variant as well. So it's dangerous to take interpretation from one person, but that is still remarkable, I think. Yeah.
She has amyloid in her head?
I don't know. I think she's died recently, so maybe Henna knows.
I have a very naive question. If A-beta immunization could prevent the accumulation of the plaque, although it doesn't clear it based on early studies, is it possible to create vaccines for A-beta for healthy people so that we can completely prevent accumulation of A-beta?
Well, that's being that's not a stupid question. That's being actively discussed. That's being actively discussed. Of course, one always has to be aware that APP and maybe A-beta itself has a function, and so one always has to be a little bit worried about doing the type of experiment that you're suggesting, but it's definitely being discussed.
Thank you.
Okay, thanks, guys. Like I say, I'm here all week. I don't know. I should have my email address. Please, I don't want 100 emails tonight, but my email address is my name and then UCL, University College London, AC, which stands for Academic Computing UK. So if anybody wants to, I'll be here, as I say. So I'm happy to meet people either singly or in small groups. I'm really happy to do that.
Thank you. So let's join to thank you, Professor Hardy. Thank you.